last time I was talking about hormones and lipid-soluble and water-soluble hormones, and ended with talking about the, very briefly, the receptors of the two types of hormones. Water-soluble hormones have receptors on the cell surface, cell membrane, whatever you want to call it, and lipid-soluble ones are inside the cell. The book has these two pictures that demonstrate this. So I want to show you these pictures, and hopefully we'll be able to see them on the screen well enough. Is that too dark?
No, it's better.
Just the lamp in this projector is pretty bad, and so I want to. So I think we can see things pretty well there. I brought it up with the department chair, and she's gonna. We were under the impression somebody put a work order in to fix that, and she's gonna check on it. So hopefully the projector will be brighter soon. But this figure shows us how a water-soluble hormone interacts with its target cell and where its receptor is. So they show the water-soluble hormone just as a triangle-shaped thing up here, moving through the bloodstream. It leaves the bloodstream through a capillary into tissue somewhere in the body, and if there's a target cell for that hormone in that tissue, it'll have a receptor on it. Now, I've always been kind of amused by this picture. The receptor here kind of looks like a pimple on the outside of the cell. But really, a receptor in a cell like this would be a transmembrane protein. It'd be a protein that's integrated into the cell membrane, and it has an extracellular and an intracellular component. The extracellular part of the receptor, what's sticking outside the cell, is what the hormone binds to. And then on the inside of the cell, it interacts with a second protein called a G protein. I've been kind of bad about doing this as I've been recording these lectures, but I really should, instead of, well, I guess I haven't been doing it because it's hard to see it on the screen, but maybe with all the lights out, it'll be better. I should be typing things up on the screen so it's captured in the recording rather than writing it on the board where only you guys can see it. Anybody watching this misses it. Let's see. So the protein, I mean the receptor that I'm talking about for the water-soluble hormone is called a G protein coupled receptor, or you may see that abbreviated GPCR. And it's composed of three parts. The first part is that receptor protein, which is the transmembrane thing that binds to the receptor on the outside. And then that interacts with a G protein. Oops. And the G protein, and I'll go back to the picture in a little bit to show you this stuff, but I want to write this down on the board first. The G protein is an enzyme that hydrolyzes GTP. That's why it's called a G protein. You might notice GTP sounds a lot like ATP, which is because they're two molecules that are different but the same kind of molecule. The A and the G part of either molecule is the different part, but then they both have TP in their names, which stands for triphosphate. ATP you're familiar with already as being hydrolyzed like myosin does that in skeletal muscle, to power contraction. The chemical energy represented in the bonds between the phosphates in ATP can be harnessed by cells to do work. And so the G protein does the same sort of thing, just with GTP instead of ATP. And when that happens, the G protein gets chemical energy out of GTP to physically move from where the receptor protein is over to another component called the effector protein. Now, the reason I wanted to write this on the board here before explaining it all in the picture is because in the picture, they don't actually use the word effector protein. The picture is an example of a specific type of G protein coupled receptor, and it's giving us a very specific example of the effector protein. There are other examples out there. The one that the book uses just happens to be the most common one, so that's why they put it in here. But there's 3 or 4 different possibilities. And so the G protein in the picture kind of looks like a little bow tie underneath the pimple I talked about. The arrow pointing away from it just shows that when it hydrolyzes GTP, when it's activated, it physically moves over to interact with another protein. And that other protein is the effector protein. Again, in this picture, they're giving us a very specific example. They're not calling it an effector protein, but the example they're giving us is this thing called adenylyl cyclase. The ASE ending on that name tells you that it's an enzyme, and effector proteins usually are enzymes. This particular enzyme cyclates or modifies a molecule to have a circular structure. No better way to explain that without going deep into the chemistry behind it. And the molecule that does that too is referred to here as adenylyl, which is a really weird name for a molecule, but there's a historic reason why it's named that, which I don't want to go into. Adenylyl basically is the A part of ATP, sort of. Now, G proteins can hydrolyze GTP. When the G protein attaches to adenylyl cyclase, the effector protein, it will then hydrolyze ATP, which is not a real big step for it because it's a very similar molecule. But when it does that, it doesn't just remove one phosphate from it, it removes 2, resulting in AMP. So if the TP in ATP is for triphosphate, the MP in AMP is for monophosphate. And that's what adenylyl is basically referring to, that single phosphate version of ATP. The C in front of AMP there is referring to the fact that the enzyme adenylyl cyclase has done its job and made it a circular or cyclical molecule. Again, don't worry about what that really means. It's just the name. This isn't a chemistry course. We don't really care about it, but it sort of takes one part of the molecule and bonds it to another part of the molecule, just making it a circular kind of molecule. In the description here, CAMP is referred to as a second messenger. Now, yes, what I just said doesn't exactly match what's typed here. I've never ever seen anywhere else refer to it as a secondary messenger. It's a second messenger. Now, this is a chemical that's inside the cell now that will start causing changes. The example again they're using here is another enzyme is modified or activated by CAMP. And that enzyme will start doing things to change other things in the cell. If CAMP is the second messenger, the thing inside the cell causing these changes, what's the first messenger?
ATP?
No, a little further back.
Receptor protein? No, no.
A little further back. Right, the hormone. Okay. The hormone, the water-soluble hormone, can't get into the cell. All it can do is bind to the receptor. When it binds to the receptor, G protein hydrolyzes GTP, which activates it so that it can interact with the effector protein, which in this case ends up making cyclic AMP. CAMP is made because the hormone binds to the receptor. Sort of like the receptor can't get into the cell, so instead it causes things to happen inside the cell. Okay? It's like if you lost your keys and you got home and you can't get into your house, so you're knocking on the door and you're telling the dog to go find the keys and open the door, whatever. You can't get in the house, the dog's inside the house, it doesn't change. Dog's second messenger, you're the first messenger. Weird example, I know, but this can't get into the cell, so what's going to cause changes inside the cell is the second messenger, and it's only there because the first message got there. Now, we don't really call the hormone the first messenger, but calling something a second messenger suggests that there has to have been a first one there. Okay. All water-based, or water-soluble hormones use this kind of receptor system. It's also referred to sometimes as a metabotropic receptor because it causes metabolic changes in the cell. It causes chemical reactions to take place. It can also be referred to as an indirect action receptor because what binds to the receptor doesn't go any further than that. Everything else that happens is indirect because of that binding. So now, having included these other names for it, do those names remind anybody of anything? And this is more directed at people that had me for ANP1.
I didn't have it for AMP1, but to me it looks like pertaining to the metabolic.
So that's the definition of it, yeah. But actually what I'm getting at, and not having me for AMP1 means that you wouldn't have done this. Back in the book, chapter 12, which is the nervous tissue chapter, where action potentials and all that was described, there's also an explanation of neurotransmitters. There we go. Where it talks about, and here it introduces two different possible types of receptors. Direct action and indirect action receptors, or ionotropic and metabotropic receptors. There are some problems with this picture, which I'm trying to get the publishers to fix, but when the signaling molecule, in this case, or neurotransmitter, because we're in the nervous system chapters, when it binds to the receptor opens up and allows ions to pass through the membrane, depolarizing the membrane. This is sort of the initial type of receptor that you learned about. It's the acetylcholine receptor at the neuromuscular junction, what we now know as the nicotinic receptor. And then it's also possible that receptors can work this way, where the signaling molecule binds to the receptor protein, which activates a G protein, which then will, G protein, which interacts with a effector protein, which makes a second messenger, which then has effects in the cell. Again, this is the nervous system chapter. So we're concerned with depolarizing membranes. And so we do that by allowing ions to cross through the membrane. And so a G protein coupled receptor, what we just saw for hormones, causes metabolic changes in the cell when it's attached to it, or when it's activated by a neurotransmitter, when it's that kind of receptor, it's usually going to directly cause ion channels to open. Okay. So again, back to just the overall thought of signaling molecules that I started with introducing everything yesterday. The receptors are what make the target cell, something happens. Neurotransmitters generally cause ion channels to open and membrane potentials to change. And it can do it either directly through a ligand-gated ion channel, which is the top picture there, or a second messenger, G protein coupled receptor, which ends up opening an ion channel. For hormones, again, water-soluble hormones, they only use that metabotropic or G protein coupled receptor. The difference between two types of receptors is really a matter of time, how long it takes these things to happen. This receptor going through the receptor protein, the G protein, and the effector protein to make the second messenger to cause the changes in the cell, that takes hundreds of milliseconds to happen. A ion channel that's directly opened by a neurotransmitter, that can happen pretty much instantaneously, measured probably in nanoseconds. So sometimes neurotransmitters need to have an immediate response. Okay? If you touch a hot stove, you want to immediately pull your hand back. You don't want to have hundreds of milliseconds of delay allowing tissue to get damaged. You want it to immediately cause a change. But hormones and other certain neurotransmitters sometimes don't need that much of a speedy response. 100 milliseconds isn't a big deal in the greater scheme of things. You can't consciously perceive 100 milliseconds at that fraction of a second, or it's very hard to perceive. You wouldn't see something that happens in 100 milliseconds, really. For you, consciously, it would almost seem instantaneous. But at the cellular level, that kind of delay between a few nanoseconds and 100 milliseconds can mean a big difference between, like, damaging tissue or removing your hand quickly. So neurotransmitters sometimes need an immediate response, but hormones, 100 millisecond delay for all this stuff to happen isn't a big deal. Plus, this type of receptor system is very versatile. When it's activated by neurotransmitters, those types of receptors usually cause ion channels to open. But using the same basic machinery inside the cell, what this molecule does inside the cell can be tailored to what the target cell needs to do. So it might activate some enzymes that do things in the cell. It might go in and cause the genes in the nucleus to be transcribed and then translated into proteins, whatever. There's a whole bunch of possible metabolic changes that can happen in a cell with this type of receptor system. When it's that instant ion channel opening because something bound to it, all you can do is you can change the membrane potential. Okay? And hormones often do other things than change membrane potentials. But certain neurotransmitters need that to happen instantaneously, so it's a better type of the better type of receptor system to use. So again, these are water-soluble hormone receptors. There are variations on this. This example with adenylyl cyclase is not the only way that this type of thing can work, but it is probably the most common type of G protein coupled receptor effector protein. So that's why the book uses it. Now, the other picture from this section in the book, very similar, shows the lipid soluble hormone receptor, which is very different. It's inside the cell. So again, they have the hormone traveling in the bloodstream. What's wrong with this picture? What's the only thing I've said about this picture? Point out the hormone. And then I asked you what's wrong. So the big hint is it's something about the hormone in this picture that's wrong.
It's not supposed to be inside the cell, or it's just signaling.
We haven't gotten to the cell yet. Well, I point up here. Well, it's supposed to be in the blood, but it's not supposed to be
It's
freely.
supposed to be wrapped in like a protein.
Yeah, there's supposed to be some sort of transport protein. For whatever reason, they didn't put that detail into the picture, which is fine, but I do want to make that point, that if it's a lipid-soluble hormone, it will be attached to something that helps it dissolve into the plasma of the blood. But it gets out of the plasma, it gets out of the blood, moves into the tissue, and being lipid-soluble, it can dissolve into the cell membrane, which is lipid. It gets into the cell, and then its receptor is what it interacts with inside the cell. So you're probably used to receptors being the things that we've been talking about so far. On the cell surface, something binds to it, like a neurotransmitter or hormones even. But lipid-soluble hormone receptors are very, very different. They're just proteins in the cell that bind to the hormone, and they form what's referred to as the receptor hormone complex, or just a complex of molecules that are acting as a single thing. They tend to bind to DNA in the nucleus and increase gene transcription, which will then lead to an increase in protein translation. Now, it is possible, some variations here, just because something binds to the DNA doesn't mean that it's going to automatically increase gene transcription. It could just as easily decrease gene transcription. So it might bind to a gene that's normally active in a cell and quiet it down, or something like that. Probably more often it's going to be what we see here. Now, a good example of this sort of thing, the picture that we have here, this kind of egg-shaped blob, doesn't suggest what type of hormone it is, but let's pretend that this is a steroid hormone. And let's pretend specifically it's testosterone. Testosterone gets into the cell, binds to its receptor, and as a complex, that binds to the DNA. A target tissue of testosterone is muscle tissue. And when testosterone binds to its receptor and that acts in the muscle nucleus, it increases the transcription and translation of muscle proteins like myosin, actin, tropomyosin, troponin, those things that you learned about last semester. Making more of those proteins means that you're going to make more of the myofibrils inside the muscle, making the muscle fiber thicker and able to contract more. That's what's happening at the molecular level. What we would think of sort of at the organism level is that you're adding muscle mass to through the action of testosterone. That's what testosterone does normally. That was one of the things that testosterone does normally. Testosterone and analogs to it, molecules very similar to it, can be used to force an increase in muscle mass. Okay. That's what people mean when they talk about athletes abusing in performance-enhancing drugs or using anabolic steroids.
Like genotropic.
I don't know the names of all of them offhand, but so there might be some performance-enhancing drugs that work in a different way, but the testosterone analog ones use this kind of system to increase muscle mass growth by telling the cells to make more proteins. There are a lot of different ways of enhancing performance that might not have to do with hormones, but the anabolic steroids that do that are basically acting like testosterone. Testosterone is an anabolic steroid. It does that. Yes. Question or stretch?
Question. So is your production of testosterone?
Yes, I believe that's true to say. when I think of when you're working out and you're building muscle mass, what you're doing to the muscles is you're actually causing micro tears in the muscle structure. And so the muscle cell repairs itself. And I believe testosterone will aid with that muscle repair and cause those muscle proteins to be made. So people that are using anabolic steroids to increase muscle mass will take that, go work out, and then the normal repair procedures that will increase the muscle mass a little bit when you work out is just amplified because you have a whole bunch of anabolic steroids on board and it increases the response. So, yeah. Other questions? So other steroid hormones might work under might cause different changes, but for the most part, they're going to do something like this, binding to the DNA and having an effect on gene transcription and maybe protein translation. Another type of lipid-soluble hormone that we want to consider is the thyroid hormone, T3, T4, those things. And they're going to do the same thing. So if this little egg-shaped blob were instead T3 or whatever, it would get in, bind to its receptor, and it would bind to DNA and cause changes there. The thing about thyroid hormone is that it's concerned often with increasing metabolism, okay, which is getting energy out of nutrient molecules and using that energy to do stuff. Where do we harvest energy out of those nutrient molecules? Primarily in the cell. What part of the cell makes energy? Yeah. So thyroid hormones don't necessarily go into the nucleus of the cell. They might go into the mitochondria. Mitochondria have their own DNA. They're kind of like little bacteria that live inside your cells. And that's one of the theories of where they came from. Not that you have a bacterial infection. It's just, forget it. They have their own DNA. That's the point. And so the thyroid hormone receptor complex can go into the mitochondrion and change transcription and translation of the mitochondrial DNA, which will increase the activity of making ATP. It'll also go into the nucleus and cause changes in other genes in the nucleus that have to do with metabolism. Okay, but if we add a little bit onto this picture for the sake of the thyroid hormones, we could say that sometimes they go into the mitochondria and have the same effect there. Okay. That's lipid-soluble hormones. Okay. So again, these two pictures here under, where is it? Pathways involving intracellular hormone receptors, just showing lipid-soluble, and then the title there before the lipid-soluble picture was intracellular hormone receptors. This says cell membrane hormone receptors. Another way of thinking about it, lipid-soluble gets inside the cell. Water-soluble stays outside the cell, so it interacts with the cell membrane. Now, this picture is kind of a general example of how hormones kind of interact with the body. The exact example that's being used here, just so that the pictures can mean something, is about the hormone cortisol. They don't actually say cortisol in this picture because there are technically a family of hormones that would be kind of like this. And we'll talk about that family of hormones some other time. But we're regulating homeostasis. So if there's an imbalance in homeostasis, there will be some sort of sensor somewhere. Okay. And so the example they're using here is, well, they say hypothalamus glucocorticoid sensor. So there's a cell that's in the blood vessels passing through the hypothalamus that can sense the concentration of these hormones in the blood. And when that goes down, that'll cause the hypothalamus to release a hormone called CRH. That's all we have to use to refer to it. That stands for corticotropin releasing hormone. CRH will then be released out of neurosecretory cell axons into the primary capillary plexus of the hypophyseal portal system, something that I mentioned in lab yesterday and would have mentioned in lab on Monday if we had lab on Monday. Oh, by the way, for the Monday lab people, none of whom came to lab yesterday because it was snowing again, for sure. I did record the lab, so if you want to go in and hear a fresh recording relevant to this semester's class, it's in the YouTube channel for the class. But, so CRH is released into the bloodstream right here and goes down to the anterior pituitary where it finds its target cells oh wow, they don't actually put that in here. So the target cells in here release a hormone which is not named in this picture. It's ACTH is its abbreviation, which then travels through the bloodstream of the whole body and goes to the adrenal cortex where it causes the release of glucocorticoids, which is a type of steroid hormone, which includes cortisol. Now the concentration of glucocorticoids in the bloodstream is higher, and those sensors that originally got everything started over here will stop sending a signal to the hypothalamus or stop telling the hypothalamus that the levels are low, and the hypothalamus stops releasing CRH, which is the negative feedback signal that shows the hypothalamus everything's back to normal. The glucocorticoids, or cortisol is the specific example I mentioned, do other things in the body, which we're not going to get into. This is just looking at how the release of hormones can be regulated actually by a few steps using hormones. They don't actually have all the hormones listed here, but CRH is released from the hypothalamus, which causes the anterior pituitary to release another hormone, which I named as ACTH, which then has an effect on the adrenal gland. That step there, the ACTH thing, is not in this picture exactly. They just say triggers, cascade that triggers adrenal glands. So there's a series of signals, CRH, ACTH, all that gets around to making the adrenal gland do what they're going to do, what it's going to do. Okay, and that is a homeostatic balance mechanism that's regulated by negative feedback. As the hormones being released from the gland out in the periphery, that signal gets back to the hypothalamus, and the hypothalamus stops that cascade that got everything up and running before. Yeah.
So the CRH is produced by the hypothalamus if what is too low?
When the glucocorticoids are too low. So it doesn't quite show it this way in the picture, but these hormones here that are being told to be released, when there aren't enough of them in the blood, that's when the hypothalamus says, okay, tell the adrenal cortex to release it. So the imbalance is the low concentration of that particular stuff, the glucocorticoids in the blood. And that causes CRH to tell the anterior pituitary to tell the adrenal cortex to release them. Okay. So this example is actually a fairly straightforward one because the signal is actually the presence of the hormone in the bloodstream. Okay. There could be other examples here that are a little bit more complex because instead of just a hormone sensor, the sensor there would be blood pressure or something like that can respond. But the book is using this, I think, as an example because it is so straightforward. It's just the glucocorticoid levels are low, so the hypothalamus responds by telling the anterior pituitary to tell the adrenal cortex to make more glucocorticoids. And it goes back up. So. Um, feedback loops like this and some slightly different ones are not uncommon in homeostasis or endocrine function. They don't necessarily all involve the hypothalamus. And I think this next little last section, this part of the chapter, are just kind of saying that sometimes it's all maintained within a particular gland, and the hypothalamus and anterior pituitary might not be involved. Okay. In lab, I talk about this a little bit more, and again, for the Monday Lab people, I did record what I did yesterday, so you can go back and see this. But it talks about I talked about the relationship of the hypothalamus to the two parts of the pituitary, which are explained here. I don't think in lab yesterday, did I talk about growth hormone at all?
You mentioned it, I think, once.
Okay, yeah, we'll count that as not talking about it, because there's a whole story about it. Oh, before I start showing you this stuff, let me just point out in the chapter here kind of what I'm doing. I'm going through some things in the chapter. Oh, of course, you can't read it because the projector doesn't work. But the first part was an overview of the system, which is basically where hormone was introduced as a term. In the book, it also mentions glands that are commonly listed as part of the endocrine system. And then it goes into detail about the hormones. That's where I discussed lipid-soluble, water-soluble, and all that sort of stuff, and receptors. Now we're in the section that you definitely can't read because it's light gray on the screen. That's the hypothalamus and pituitary. So what I just showed you, that feedback thing, was supposed to be kind of a general example of it. How the hypothalamus and the anterior pituitary regulated a particular hormone release. Part of this, the stuff that I described in lab that I skipped over, explain the relationship between the hypothalamus and the pituitary in a little bit more depth. So if you weren't in lab yesterday, you can watch the recorded version of that. Then the next part here goes into the story about what growth hormone is. And it's a good example for a bit more complex relationship between the hypothalamus, the anterior pituitary, and a endocrine tissue out in the periphery. And so I want to talk about that story a little bit more. After that 17.3 section, which is what we're on in the book here, the rest of the chapter kind of goes through the various major glands. And a lot of these I talked about in lab. So the first two are thyroid gland and parathyroid gland. They each get their own section. As far as the thyroid hormones T3 and T4, most of anything that I want to say about that I already have, mostly talking about the lipid-soluble hormone receptor just then, and the mitochondria. But then also in the thyroid gland, there's calcitonin, which helps regulate calcium along with the parathyroid hormone. So those two together kind of match up. And I talked about that in the lab. And then there's the adrenal gland, and we looked at that in lab, and so I talked about that. That's really more about the different regions and what types of hormones come from that. And then there's a section on the pineal gland. Did I mention the pineal gland on Tuesday? Did I say anything about melatonin? Yeah, you said melanin, and I corrected you on that.
Yeah.
Right. That's all I say about the pineal gland. You're welcome to, well, actually, I have one sentence to add about that when I talk about growth hormone, but you can go to the pineal section of the book to see more of it. That's all it does, it releases melatonin. Where are we? Then it has a section on the gonadal and placental hormones. And we'll talk about those hormones in the reproductive system at the end of the semester. Then there's the endocrine pancreas, which I talked about in lab, but I actually want to touch on a little bit before I go into growth hormone. It's really hard to read some of these things. Then there's a list of organs with secondary endocrine function, which essentially there just says things like the heart and the kidneys and the digestive system, et cetera, also release hormones. And as far as I'm concerned, we're going to talk about those as we get to those particular organs in the various organ systems. So there's that. And then the last section of the chapter is about development and aging in the endocrine system, which I don't talk about, I don't deal with in this class. So you're welcome to, well, I would suggest you read that because there's a course compass set of questions that go with it, but I won't ask any questions in the graded material from that. So I want to talk about growth hormone, but before that, I actually want to get into the endocrine system, into the pancreas a little bit, which I did talk about in lab. So as far as talking about the pancreas and the tissue and all of the players deal with the lab material. When I talk about it in lab, I didn't use this picture from the book. So I do want to point this out and just, one, review what I said in lab for people that were there yesterday, and to introduce this to the people that weren't there yesterday, so that you have this to understand something about growth hormone. So this is a kind of figure-8 model of how blood glucose levels are regulated. Okay. So in the middle, this is talking about when our blood glucose levels are normal, our homeostatic set point. And there's a loop going down from here and a loop going up from here, which is when blood glucose levels go down, this happens. When blood glucose levels go up, this happens. I thought it's very clever in this book, for this particular example, to graphically represent the blood glucose levels. They're using a little meter that somebody with diabetes would use to check. Okay. So this, if you have to check your blood glucose levels because you're diabetic, you'd have one of these things. You get a little drop of blood on your finger from a pinprick, basically, and then that drop of blood is assessed by this meter, and it gives you a number that's the blood glucose levels. Yeah.
My son is actually growth hormone deficient, and he has to do this when he wakes up.
When he wakes up.
Mhm.
Okay, so we'll kind of touch on why that might be true for him, but that's a fairly rare condition.
Yeah.
Diabetes is more common. So here they have a picture showing a normal blood glucose level. They're saying 90 milligrams per deciliter, and then 50 would be low and 160 would be high, just to represent those changes in blood glucose levels. And I just think this particular picture is kind of inventive way of depicting that in graphic format. So let me zoom in on the bottom half of this picture, which is what happens when blood glucose levels are low. In the pancreas, so in this picture here, these little blue flower-shaped things, those are pancreatic islets, which are where endocrine function takes place in the pancreas. And again, that's something I talk about in detail in lab with micrographs and but cells in there, when blood glucose levels are low, sensing that, will release the hormone called glucagon. Glucagon targets pretty much the liver, although there are possibly a few other minor targets in the body, but mainly it targets the liver. It tells the liver to release glucose into the bloodstream to get blood glucose levels up. The liver does that in two ways, either by breaking down glycogen, which is stored glucose, so that individual glucose molecules are released in the blood, or by causing glucose to be reconstituted from the leftover parts from when it was broken down before. So that's basically what these two say. The first one says glycogenolysis, glycogen, I said it right, which needs to break down glycogen. And then the other one is gluconeogenesis, which literally means to make new glucose. So both of those are going to raise blood glucose levels. In the book here, they mention it inhibits body cells from taking up glucose from the blood. I'm not clear on that because that would require all cells to have glucagon receptors on them, and I don't think that's true. So, but I'm not an expert in the field, and there might be some evidence that this actually does happen. But as far as I know, it doesn't. Rather, passively, when there's not enough when there's not enough glucose in the blood, cells aren't going to take it up. Okay. So this might more be a passive response because blood glucose is low. But what glucagon really does is tells the liver to release glucose either by breaking down glycogen or by reconstituting new glucose molecules. And so that brings the blood glucose levels back up to normal, hopefully. The other half of this picture is what happens when blood glucose levels go up. So here we see a number representing that. And in the pancreatic islets, there's other cells that are responsible for responding to high blood glucose levels, and they release insulin, which you've probably heard of before. Insulin basically does the opposite of what the book just said for glucagon, but that last thing I said that I don't think the book's right about it inhibiting glucose uptake, that's because insulin triggers glucose uptake. All cells in your body have insulin receptors. There's every cell in your body is a target for insulin. And so I think what the book said there about glucagon inhibiting that is really just the absence of insulin means that it doesn't happen. So when you're in low blood glucose levels, there's no insulin, so your cells don't take it up. But when insulin is present, all of your cells will take the glucose up. It also, for the cells in the liver that take up that glucose, tell it to make glycogen, or you can also think of it as stop breaking down glycogen. So when the liver takes up glucose, if it stops breaking down glycogen, then it's going to store some of that new glucose back into glycogen molecules. And it inhibits making new glucose, which is actually not a very efficient way of storing energy, but it stops doing that. Tells the liver to stop wasting energy making glucose again. What's not mentioned here is insulin also will cause other nutrient molecules to be taken up. If you have glucose in your bloodstream, you likely have other nutrient molecules, lipids and amino acids available too. So while it's the stories about regulating blood glucose levels, which is all that this picture says, since you probably just ate a meal, you have other nutrient molecules, and insulin actually stimulates those to be taken up too. Just introducing that basic idea that insulin and glucagon help regulate blood glucose levels and sort of the way they do that, I want to talk about growth hormone. And the reason why I want to just touch on insulin a little bit first is because there's a very close relationship between growth hormone and blood glucose regulation. In the growth hormone story here, part of what's going on is that hypothalamus regulation of things. So the negative feedback example that I gave you earlier was a very simplistic example of the hypothalamus being involved, but it's also true for growth hormones, so let me talk about that. In the hypothalamus, there are cells that release a hormone called GHRH, which stands for growth hormone releasing hormone. That hormone travels from the capillaries right here to the capillaries right here, and the target cells for GHRH in the anterior pituitary then make growth hormone. So they're told by the hypothalamus to release growth hormone. GHRH tells it to make GH. GH then gets into the bloodstream and goes all over the body and has a number of different effects. In the book, they put these in order A, B, C here. I actually want to start with C because I think it's the most important thing that growth hormone does. And actually in C, I'm going to talk about sort of the thing that they've kind of hidden in this picture, which is down here at the bottom, it says IGF-1 release. IGF-1 is the abbreviation for a hormone that I want to talk about. And this is going to start off with, explain to you why I just included insulin, or just talked about insulin. IGF-1 is insulin-like growth factor 1. Okay. Now, the fact that it's got a 1 in front at the end of it suggests that there's more than one of these types of things, but we only care about the first one. So if I don't call it IGF-1 and I just call it IGF, that's what I'm talking about. Okay. We don't care about the other insulin-like growth factors. But now, that name implies a lot that I have to explain to you. Every animal has an insulin gene, and insulin does for all those animals exactly what we expect, roughly what we expect, which is to regulate glucose levels and metabolism of glucose. Now, I say every animal. I actually don't know if it's something like it's present in plants, but plants make their own glucose, so they don't really need to regulate glucose levels quite the same way. They don't get a burst of glucose when they eat something. They're plants. They don't eat things. They make the glucose themselves. So physiologically, plants are very different. So let's just ignore those. We'll just talk about animals. So there's an insulin gene in every animal, which is to say that the ancestor of all animals had insulin as part of its physiology. Also, all animals have a gene for IGF, right, which say that all animals have that. But understanding how genes modify themselves over the years or over generations, we can say, even though every animal alive has both of those genes, we know that at one point there was an animal that had an insulin gene, and that insulin gene got duplicated. So in reproduction, DNA is passed on from parents to offspring, and the DNA is duplicated. You know, a new copy of, or I shouldn't say duplicated. Cells are passed on, sperm, egg cells, those kinds of things, and within them they have DNA that's being passed on from generation to generation. In the production of one of those gametes or sex cells, the DNA that contains the insulin gene had to have gotten duplicated so that 2 copies of the insulin gene was passed on from one parent to its offspring. Now, the other parent would also pass on an insulin gene probably also, but that's not important. The fact is an extra insulin gene got into the genome for this animal. Insulin, since it exists in all animals, we know is very important for the success of animals. So there's a lot of pressure for the insulin gene to stay functional. It might change a little bit if you look at a human insulin gene and compare it to the insulin gene of a sea slug. They might look kind of different, but they're fundamentally the same gene. Okay? You couldn't put sea slug insulin into a human and necessarily get the same kind of response, but they're pretty similar because it's such an important gene. It has to stay pretty similar. But when you have 2 copies of insulin, it's possible for one of those to mutate because the non-mutating version of insulin does insulin's job, and the mutating version can then change. And that's where insulin-like growth factor came from. It's a variation on insulin. If you compare the 2 genes, the sequence of nucleotides that set up the sequence of amino acids to make the proteins, they're very, very similar. There's one little difference there's more than one difference, but there's one little difference in the IGF gene that alters the structure of the protein. Okay. And so it's a different protein. That new protein, that mutated protein, took on a different role. There was a function that it was able to produce, I suppose you could say, in the organism that helped that organism survive. So it passed on its mutated version of the IGF gene, and that became an established gene in the genome for its own reasons. Insulin, we just learned about, has to do with regulating blood glucose levels. IGF isn't about blood glucose levels at all. The function that it ends up being important for is it helps growth hormone cause growth. Now, I'm familiar with this because when I was in graduate school, the first lab I worked in was a neuroendocrine lab where we were studying, actually I was studying IGF's effects on the brain, but the lab in general had a whole grant that they were studying growth hormone and IGF together. And so I worked with growth hormone as a student. And where was I going with that? When I was a student, the way that we understood growth hormone was growth hormone itself doesn't actually cause growth. Instead, all of the growth that's attributed to growth hormone, why it gets its name, growth hormone, is actually handled by IGF. And the reason why we call growth hormone, even though it doesn't really cause growth, is because that's not the picture. That's not the picture. There it is. Okay. Is because IGF is released from the liver when growth hormone tells the liver to make it. Okay. So a little while ago, that negative feedback example I gave you with corticoids, the hypothalamus told the anterior pituitary to tell the adrenal cortex to release that hormone. Okay. Here, the hypothalamus tells the anterior pituitary to tell the liver to release IGF. Okay. So growth hormone is the signal that tells the liver to release IGF. And then IGF causes growth. Okay. Now, when I talk about IGF causing growth, primarily what we think of is making long bones longer. Okay. The growth plate, the epiphyseal plate between the diaphysis and the epiphysis of a long bone, which causes long bones to get longer so that you go from being a little tyke to a big tyke. That's all caused by IGF. It causes the cartilage and the growth plate to grow, and then it ends up calcifying and becoming bone. It also causes other tissues to grow along with that. If your bones grew longer and nothing else grew longer, then you'd have a lot of problems. So IGF also causes growth of muscle tissues, and nerves and blood vessels and skin and that kind of thing. So it just causes growth. And by that I really mean the growth that causes us to get taller. But IGF-1 will not cause growth unless the growth hormone signal is there to get it started. So that's why it's called growth hormone. That's what starts growth. Question?
So if you introduced growth hormone to someone who is already full grown, would it just affect like the muscles of that person, or would that still affect the bones?
No. Well, it will not affect the bones because the bones have stopped growing. But growth hormone has a number of other effects, and everybody has growth hormone in their system throughout life. Just after you've grown fully to your full height, it no longer has an effect on growth of bones and the surrounding tissues, but it still does have a number of effects, which will get and glucose regulation.
Yeah.
And stuff like that. Yeah. So that's really the important part. Oh, sorry, I'll leave.
I just want to make sure I understand it correctly. Saying growth hormone signal tells the liver to make IGF. And then when it's coupled or when IGF comes in contact with GH, growth hormone, causes the growth of the bones.
Let me try laying it out this way. So the hypothalamus, HGH is an abbreviation for hypothalamus, will tell the anterior pituitary to do something, which will then tell some other gland to do something. Okay. So in the first example, the negative feedback example, hypothalamus released CRH, which tells the anterior pituitary to release ACTH, which tells the adrenal cortex to release corticotropins, okay. Which I can write that here. So, for example, CRH, oops, causes ACTH, which causes, oops, from the, I'm just going to put ADR for adrenal. Okay. Or we could say GHRH from the hypothalamus causes a release of GH from the anterior pituitary, which causes a release of IGF. Whoops, that's not an I. From the liver, and that's what causes bone growth. Other muscles. Okay. So the hypothalamus, anterior pituitary, and the rest of the endocrine system works largely just straightforward like this, and we just plug in different examples here. Okay. So the negative feedback example was this: CRH, ACTH, corticoids, growth hormones. Story starts with GHRH from the hypothalamus, which then causes the anterior pituitary to release GH, which then causes the liver, the target of GH, in this case, to release IGF-1. And then the IGF-1 is what primarily causes growth. Does that answer the question?
Yes.
Okay. So back to the picture, because that's not all that growth hormone does, but partly because of my experience working in that growth hormone lab, and it's called growth hormone. Obviously, growth is the reason it was named. Why wouldn't we talk about that primarily? And in A&P1, you learned about bones and growth plates and all that stuff. This is what causes it. Okay? I'm amazed with pretty much every textbook that they just fail to reinforce that. This is a prime place to tie an A&P1 concept to an A&P2 concept. They never do. But that's what's actually going on here. Now, what growth hormone does, besides this, is what's also represented here. So I actually, again, do want to start in the C part of this. The rest of what it's saying here is here called the diabetogenic effect, which sounds kind of weird. I've also heard it referred to as an anti-insulin effect. Growth hormone will stimulate the liver to break down glycogen into glucose. What does that sound like to you? We just talked about this a little while ago. Telling the liver to break down glycogen. Not insulin, but it's the other thing. Insulin and glucagon. Right. Glucagon tells the liver to break down glycogen into glucose. That brings blood glucose levels up when glucagon is involved. So growth hormone actually pretends to be like glucagon. Okay. So it tells the liver to release glucose and to make IGF. The reason why it does those two things together is because IGF is insulin-like. Okay. So I told you the thing about the duplicated genes. What I left out of that, because it just sort of makes that part of the story a little more complex, is that it wasn't just the insulin gene that got duplicated. It was the insulin gene and the insulin receptor gene. There is an IGF gene, which is a mutated version of that copy of the insulin gene. But there's also an IGF receptor gene, which is a copy of the insulin receptor gene that got mutated. They kind of went together, things changed. So when IGF has an effect, it has an effect because the cells it's targeting have IGF-1 receptor. But because IGF is insulin-like, it can also bind to the insulin receptor. The thing is that when IGF binds to the insulin receptor, it's about 10% of as effective as insulin. Okay? So insulin binds to the insulin receptor, it causes blood glucose levels to drop, and it stops glycogen breakdown by the liver, et cetera, what we talked about for insulin. Insulin-like growth factor can do that too, but about 10% as effectively. So not as much, but it still does have an insulin-like effect. So to offset IGF having that effect, growth hormone will pretend to be glucagon, okay, to offset the insulin stuff. IGF causing growth will mean that growing tissues need energy. Okay? And if IGF stimulates growth and stimulates non-growing tissues to take up glucose, it'll be counterproductive. The growing tissues need energy. So growth hormone counteracts the insulin side effect of IGF by acting like glucagon. Okay. That's actually not all that it does to help growing tissues get energy. It also will affect adipose tissue and tell it to break down lipids, because lipids can be used instead of glucose to make energy also. So between the glucagon-like things and the effect on adipose tissue, it provides the energy necessary for growth. Okay. Now, I'm standing here for a reason. One, these two boxes both help growing tissues get energy to grow. Two, this box bugs the hell out of me. Like I said, when I was first in graduate school, I worked in this lab that studied growth hormone. And back then, we talked about how growth hormone doesn't actually cause growth itself. All of the growth actually happens because of IGF. In the book here, they say growth hormone has growth effects, okay, which is contrary to my understanding of growth hormone. However, my understanding of growth hormone is a couple of decades out of date. So when I was in graduate school, we did not consider growth hormone to have any direct growth-causing effects. It is entirely possible that since I was working in that lab and studying growth hormone, that new things have been discovered, and it might be true. I doubt it, and Casey does too, apparently, just because part of establishing that understanding before was to test growth hormone's effects, to add growth hormone to growth plates and bones, and directly it caused no growth. So I really don't believe this, but I'll give the book a little bit of leeway that maybe new things have been discovered that make this box truer than I believe. But really, I think this box should solely be IGF still. Thank you. In the end, it's not that important whether that's true or not. The point I'm trying to make here is growth hormone causes IGF release, which causes growth, but IGF has a side effect of lowering blood glucose levels. So growth hormone also acts like glucagon to keep blood glucose levels higher, as well as having an effect on adipose tissue, which glucagon doesn't do itself. Okay. So back to John asking, well, what about adults? And Casey mentioning the blood testing that
My son.
sorry, I was about to say daughter, has to do. That's because growth hormone actually does play a role in regulating blood glucose levels. Okay. As an adult, that's about all it does. Okay. As an adult, growth hormone doesn't really cause any growth because we're pretty much full grown. There are some growth processes that can still take place in adults, not bone growth so much as, so it could be, yeah, if you're working out, you're building muscle mass, it might play a role in that. Also, IGF has an effect on the brain. It can increase neuron growth and change and that sort of thing. Actually, my dissertation in graduate school was studying the effects of IGF on the brain. And what I proved is we don't know. I did all this work in graduate school, published a dissertation, et cetera, and my dissertation basically said, we don't know what IGF does in the brain. Another lab had proved that you could give rats IGF, and as they got older, they still performed in maze tasks the same way that juvenile rats did, compared to regular old rats that couldn't do as well. So I went in to look at the brain and figure out why that happened. Got absolutely no results, which is just the things that I looked at were not what IGF was changing. But it does have an effect on the brain that it makes aging brains be able to do things a little bit better.
Has the effects of IGF damage nerve tissue shown any improvement?
Not that I know of. And again, a lot of that has to do with the fact that I'm not in that research stuff anymore. So if anything's been discovered in the last couple of decades, I don't know about it. But from what I was involved in back then, it was actually looking at normal rats. So you can think of aging as being a damage process. And so yes, it did improve that. But we didn't look at it, what it did to actual brain damage situations, like a spinal cord injury or something like that. I don't know that it has any effect that way, but it can keep brains acting younger for longer, or something like that. And that's going to come from growth hormone. Now, to finish up that idea, let me go back to the growth stuff for a second. Does anybody know when your body, when you were a child and adolescent, when was your body actually growing?
12, 13.
Not year-wise, but on a day-to-day basis, when was growth taking place?
You slept?
Yeah, it's largely happening at night, okay? Which is generally when people are asleep.
That's when they give you the meds.
Okay.
When you're right before that.
Now, when I was a kid, I heard that and I thought, hey, I'm so tall because I slept so much. It's not how it works. Actually, what it is growth hormone releasing hormone and growth hormone released because of it are released at night. The lab I worked in, I was studying IGF, so I was doing other stuff, but they also were studying what's called the pulsatility of growth hormone, which is when do growth hormone levels increase? So we had to sample tissue to look at how much growth hormone was there over a 24-hour period in the whole population of subjects. And so we saw in the species we were studying when that happened, and then other labs doing similar work established that in humans, growth hormone pulsatility presents itself as 2 increases in growth hormone levels overnight. Okay. So growth hormones released and at night causes all these things, and actually IGF acts as the negative feedback signal, just like glucocorticoids do. So when IGF levels go up, that causes growth hormone levels to stop to go down, which stops releasing IGF. But then it's still nighttime, so then it's enough time for a second wave of growth hormone to be released. But then during the day, there's not a whole lot of growth hormone. And that allows for growth processes like I described to take place at night. But now you're all grown up, but your body's used to releasing growth hormone at night, which is useful because of what growth hormone continues to do for humans. So growth hormone acts like glucagon. Initially, we described it to offset the insulin effect of IGF, but it also is helpful because of when growth hormone is present. When are your blood glucose levels lowest?
In the morning.
In the morning, when you first woke up. Why? Yeah, you haven't eaten anything in,
A certain amount of time.
let's hope, 8 hours. Or if you have a kid, maybe 5 hours. Anyways, the point is you've been fasting and your blood glucose levels are as low as they can be because you haven't been taking anything in. And you want to keep those as high as possible, so glucagon's struggling to keep up. But to help glucose out overnight, you have growth hormone release. Okay, it's nighttime, growth hormone's released. Growth hormone causes the liver to break down glycogen to keep blood glucose levels high. So it helps keep blood glucose levels from completely bottoming out overnight because of when it's released and the fact that it acts like glucagon. That's largely what it does for adults, is this glucagon-like effect. So, and then again, like I was saying, it does cause release of IGF-1, which probably continues to help with certain tissues like nervous tissue or maybe muscle tissue to continue going through some growth processes even in adulthood. But it's not going to make people keep growing taller, and that stops at 15, 20 years of age. So that's basically growth hormone. Okay. This is, of course, the book's picture. The important parts are the A and B, A and C panels here, and then growth in the middle. Like I said, I don't quite believe this is true, but I'm not familiar enough with the science to say that that's out of date, but that's true. Let me just hold on a second. Let me just make sure I'm not skipping anything. Yeah, that's pretty much it. So again, if you're in the Monday lab group and you want to listen to the recording of what I did yesterday for the Wednesday lab group, go into the syllabus, and right after the course schedules for the Monday and Wednesday halves of the class, there's a section here that says online resources, and the link here in the sentence all by itself in the middle says YouTube channel. Click on that, and right here it says AMP2 endocrine tissue lab. That's what I recorded in lab yesterday. So if you'd like to, if you were in lab yesterday and you want to watch it again because we're just so excited and you gotta relive it, go ahead. But if you're in the Monday group, that's what we did yesterday that you could have recorded. Otherwise, have a nice day. Sorry for the couple of minutes over time.